Welcome to my channel, guys. Today I'm going to discuss pancreatic cancer. It's part of my lecture series on this channel, and I'm going to try to discuss certain topics that are of interest to me and hopefully are of interest to you guys. So let's get started. Pancreatic cancer is a very deadly cancer. It is considered the fourth leading cause of cancer-related deaths in North America. In Canada, there's about 4,700 cases that are diagnosed every year, and about 4,300 die from the disease every year. Now, in the U.S., you can just time these numbers by 10. So let's say maybe about 50,000 that are diagnosed and about 50,000 that are die from it. So those are pretty big numbers. And the reason why these numbers are big is not because the disease is common. In fact, it's considered a relatively uncommon cancer. The reason why is because of the low survival rate, which is about 4% and in some cases 3%. So we're talking about the 5-year survival rate. So what this means is that if you have 100 patients that were diagnosed with the disease today, about 5 of them will survive. About 4 of them will be alive 5 years from now, so in 2024. Those are very slim chances. Now, from a biological perspective, the reason why pancreatic cancer is this much of a deadly disease is because of its aggressive and invasive nature and its ability to metastasize into other parts of the body. An example would be to the bone, to the brain, and even to the liver. Now, from a genetic perspective, as scientists, we do know, at least in 95% of cases, what's the causing genetic event, and it's a mutation in a gene known as KRAS. A single change in the nucleotide or the DNA sequence will start or initiate the cancer, which in turn will then accumulate more mutations. A series of mutations are actually well understood, and by undergoing these mutations or gaining these mutations, this cancer will actually become a full-blown cancer and metastasize and lead to patient death. Now, when it comes to how we treat pancreatic cancer, it's quite an outdated regimen. So usually when a patient comes in is suspected of pancreatic cancer due to showing symptoms such as, let's say, jaundice, losing weight, not being fatigued all the time, older in general, they'll suspect pancreatic cancer. They'll biopsy the disease potentially. Well, they will biopsy the disease, and they will decide whether it's a local disease or it's a metastatic disease. This is a very clear distinction, because a local disease, which means it's just stuck or confined to the pancreas, means that these patients can undergo surgery. This usually increases their survival rate. However, if they suspect that this cancer is metastatic, that is, it has gone to the liver, to the bone, which could be confirmed through imaging, then these patients cannot undergo surgery. In fact, if you do take them through surgery, they will likely die from complications from the surgery, and if not, the surgery usually makes it worse, makes the metastases worse. So the disease that was found, let's say, in the liver is considerably worse after the surgery because of the invasive nature of surgery. And you can't really take out metastatic disease from the liver. So you can take it out, let's say, from the pancreas, sure, but when it comes to different sites, it's virtually impossible to do that. And so surgeons don't resort to that route unless it was purely for palliative care, or at least for improving the well-being of the patient. Now, if you have metastatic pancreatic cancer, the only option would be chemotherapy, long-term chemotherapy, which is followed by follow up, and the patient is monitored closely to track the progression of the disease, whether it's it got worse, it got better, or was a stable disease. If it's local disease, then it means that the cancer is potentially resectable. Patients can go under, and the tumor can actually be taken out. Now, in some cases, some patients will receive chemotherapy right after they finish surgery, just to make sure everything is kind of all gone and make sure that nothing comes back. Now, there are in some cases where you have a locally advanced disease where the risk of leaving some cancerous tissue inside is relatively high. And so instead of just sticking to surgery, they also use radiation. So they will take out the tumor area, but they also radiate the surgical margins, or the areas around the what used to be the tumor, just to make sure they kill everything. And for the longest time, we know that resection improves the survival of these patients. And so if we can catch the disease early, identify it as a local disease, then we can actually resect it and dramatically improve the survival of these patients. Now, I'm going to get a little bit scientific here, but I just want to explain to you what's the pathology of pancreatic cancer. How does the disease start? When you have a normal tissue on the left-hand side, that's what a normal pancreatic tissue looks like, and how it goes through various phases of disease, known as PAN-IN. So PAN-IN 1A, 1B, PAN-IN 2, PAN-IN 3. Those three stages are actually what we call precancerous lesions, and they get worse as you go from 1A to 1B to 2 to 3. And you can see that if you look closely at these ducts, you can see that the morphology, or the shape of the way this tissue looks, is less, looks nastier. It's less representative of the original tissue. Normally, in a normal tissue, you'll see in a pancreatic duct shown here, you'll see a single lining of cells known as the epithelial cells, and they have a certain shape, usually cuboidal shape or cube-like structures. And as you move along through these lesions, precancerous lesions, more cells are there. They're not a single layer anymore, and they just look like all over the place. You can tell that there's what we call an uncontrolled growth, and there's loss of differentiation. So the cells here are not behaving the same way the normal cells would do. This is still considered precancerous lesions, and the problem with these lesions is that they're not easily diagnosed. In fact, usually at this stage, the patient does not show any symptoms. You don't know that the cancer is developing, and so it is crucial that we figure out a way in the future of diagnosing patients at one of these three stages, because at that stage the disease has not metastasized, and it is possible to take out the tumor and actually save the life of the patient. Now, the last stage of the disease is what we know as the adenocarcinoma, which is the state where it's a full-blown disease. These cells has no resemblance to the original normal tissue. The duct is almost absent, and what you get is basically a series of, or a mass or tumor mass of cells that are abnormal in shape, much larger than a normal cell. They're very invasive in nature. They start moving into the surrounding tissue, and what's around the cells, what we call a stroma, is also modified heavily by the cancer cells, which usually allows these cancer cells to metastasize, to invade the nearby tissue, and to grow and finally get to the bloodstream where they can go to the bone and go to the liver or go to lungs or even the brain. Now, at the bottom here with these red and green lines, this is just a representation of all the genetic events that we've characterized that give rise to the adenocarcinoma or the full-blown pancreatic cancer. As I discussed earlier, the KRAS mutation is an early event, as you can see here. But there are also other genetic events which I'm not going to get into. But I do want to point out TP53. It's a well-known what we call a tumor suppressor. So it's a gene that's usually known to keep things in check. It keeps the cells safe in case something goes wrong. If there's a mistake in the DNA, anything wrong, it just causes the cells to die. And it's kind of like a protective mechanism. The problem is, in this cancer and many other cancers, they lose that gene, or they lose a functional copy, or they lose a good copy of this gene. And then that means that whenever a damage is happening in the cell is not able to kill itself. And so you get the progression of these abnormal cells that are not dying, and combined with the effects of the other genetic variants, you give rise to cancer. So finally, just kind of like a summarize what I've just talked about and kind of put things in perspective. You get at the beginning in step 1 here, cells are growing within the duct of the pancreas, just expanding, making more copies of themselves, what we call uncontrolled growth. They're acquiring those genetic mutations, the KRAS mutation, when at some point they will acquire specific mutations that allows them to break away, what we call a process of invasion. At that point, again, another series of mutations will allow them to not only metastasize, not only leave the duct, but actually metastasize into the bloodstream to go to the brain, to the lungs, as I said, liver, bone, and even kidneys. But when it goes to these tissues is when it sabotages their function. Now you've got disease that, pancreatic cancer cells in a tissue that's not supposed to be there. It's growing, it's affecting the function of the cells that are there. And slowly these tissues start giving out and basically cannot maintain their own functions, and the patient succumbs to the disease. Thank you for watching, and see you guys next time.